So yeah, you can start, Liron.
Thank you, Laura. Hi, everyone. So as Laura said, my name is Liron Zahavi. I'm a PhD student at the Weizmann Institute of Science at the research group of Professor Liron Zahavi. I'm a computational biologist, and I study the associations between the human gut microbiome and our health. And okay, so the high-level goal of everything I'm going to talk about today is understanding better how does the human microbiome, and specifically the human gut microbiome, interacts with the host health. This talk will have two parts. The first one will be a short overview of common approaches to study the microbiome, and specifically about metagenomic analysis of the human microbiome. And in the second part, I will tell you about our most recent research, in which we study the interactions between the gut microbiome and health at single nucleotide resolution. Okay. So let's talk about the metagenomic pipeline. You have a study, you recruited the cohort because you want to study an association between the microbiome and some disease. Now let's call it the blue disease. So you recruited people with both with and without the condition. And you collected their microbiome samples. The easiest way to do that, to sample the gut microbiome, is through fecal samples. So you will have samples both from the healthy participants and the sick individuals. And now what? One approach is to experiment, take the microbiome sample from the sick individuals, put it into a new organism, whether it's a mice or even a person, and see whether it affects its health. And then take microbiome samples from the healthy individuals, also put it into a new organism and see whether it has an effect. This approach can help you establish causality of the microbiome in this condition. It does not, however, tell you what component of the microbiome affects this condition, or how the microbiome interacts with this disease. A second approach will be to profile the microbiome, see what's in the sample, and compare the healthy and the sick individuals and see whether you can find some component of the microbiome that can explain these differences and is associated with the disease. And to profile the microbiome, what's usually done is you have the fecal microbiome samples, and then you extract DNA from the samples and you sequence it. One approach will be 16S sequencing. It's a very common approach in which you specifically amplify and sequence a specific region of the 16S RNA gene of the bacteria. And then you have the sequence and you can align it to known 16S genes of different bacterial species, or you can use higher taxonomic level. It has some limited resolution because you can only differentiate between different taxonomic classes as much as their 16S gene is different. But this approach is pretty cheap, and it's also very effective when you sample body sites of the different microbiome body sites in which you have a lot of human DNA and not a lot of bacterial DNA. So this will have better yield. The second approach is metagenomic sequencing. And in this approach, you pretty much sequence all the DNA you can in the sample. You can see everything that's in your sample, but you need to also think about how to make sense of it. So what's metagenomic sequencing? In general, it's taking the DNA, sequencing everything. In most cases, you'll have short metagenomic reads. So you'll end up, in each sample, you'll have this list of short sequences that represents the samples. And these sequences originate from, these are fractions of the genomes of all organisms you sampled. So you'll have fractions of human DNA, you'll have fractions of bacterial DNA, fungal DNA, maybe even of the food that the participant ate that has DNA. And now how do you compare the samples of different individuals when you have this list of short reads? So again, there are two common approaches. One is to characterize the taxonomic composition of the sample, which means you'll take these short sequences, you'll align them to a reference genome of known bacterial species. And then you can say for each sample, what's the composition of different species in the sample? What's their relative abundance, presence, absence? And you'll compare the taxonomic characterization of the healthy individuals and the sick individuals. And maybe you'll find some bacterial species that are associated with the disease. A second approach is functional characterizations, which means that you'll not characterize the species composition, but the gene composition or the pathway composition in the sample, the functional characterization of the sample. You can align your reads directly to genes and then see whether there are some pathways that are enriched with being sick. And you can also combine the two approaches. Some people will first align the reads to reference genomes of bacterial species, then say that there are certain bacterial species that are enriched in the samples of the sick individuals, and then go from that, ask what genes do these bacteria have, and then say something about the functionality of the microbiome of the sick individuals. Now, maybe the approach that I'm mostly seeing in most microbiome papers that I've read is the taxonomic characterization or the combination between the two. And the limitation with the taxonomic characterization is that you first, once you have a list of bacterial species, you still need to make sense of it to find what's like hypothesize about the mechanisms, why this specific bacteria associates with disease. And also you need to think about the phylogenetic level, taxonomic level at which you want to examine the microbiome. Okay, so a brief summary for this brief introduction. Pipeline of very common pipeline for microbiome studies is you recruit a cohort, you take fecal microbiome samples, you sequence it. In metagenomic sequencing, you get small genomic fragments from everything that's in the sample. And then you need to consider your hypothesis, what you already know about this condition, about which species may be associated with it, which bacterial function may be associated with it, and then decide on the resolution with which you want to characterize the microbiome sample. Okay, so I'm moving on to the second part. Okay, so now I will tell you a little bit about our research. And you can think about it maybe as a continuous of the taxonomic characterization approach. So what's the motivation? We know that within bacterial species, there is a lot of diversity. You can take two bacteria of the same species and they'll have different functions, different genomics. One bacteria may have antibiotic resistance while the other does not. One bacteria of the same species may be pathogenic while the other is not. Maybe one bacteria got a new gene through horizontal gene transfer that other bacteria of the species do not have. So sometimes looking only at species composition is not enough because there are some differences beyond that that you miss. So just so we're all on the same page, what are SNPs? Single nucleotide polymorphisms are positions within a species genome that vary between different organisms that belong to this species. So for example, in the human genome, we know we have a lot of SNPs, a lot of positions that in which people can be different from one another. And these differences may also be associated with different phenotypes of the people. Okay. So in general, I said that bacterial species have diversity within them. And specifically in the microbiome, it's also known, this paper was already published 9 years ago, that there is a lot of diversity within bacterial species in the microbiome. And specifically, this paper found 10 million SNPs in gut microbiome species. So this level of variation definitely exists within the microbiome. Why is it interesting? Sorry? Did anyone want to ask anything?
I think someone just forgot to turn off the microphone.
Okay. So this level of variation definitely exists within gut microbiome species. And why is it interesting? So one motivation I found in this paper that was published 2 years ago by Emily Blaskus. And what they show here is that they discovered there is this Parkinson's drug that has a different efficacy for different people. It's not really understood why different people respond differently to the drug, but it has something to do with different metabolism of the drug. And what they found, among other discoveries, I recommend reading this paper, is that there is one amino acid substitution that naturally occurs within gut microbiome species. Some people will have in their gut microbiome bacteria a serine, while others will have arginine. And this difference in vitro was shown to affect the ability of the bacteria to metabolize the drug. So one small substitution in one species in the gut microbiome may explain why different people respond differently to this drug. So this is very interesting. So taking both papers together, we already know that there is a lot of diversity, a lot of SNPs in the gut microbiome, and we've seen that they have the potential to affect different people's response to drugs. So the next question will be, do all these 10 millions or whatever SNPs we have in the gut microbiome associate somehow with our health?
Jaa, aga siis on nagu need muud büro.
Yeah. So this is what we wanted to test. We wanted to systematically associate single nucleotide polymorphisms in bacteria of the gut microbiome with different health factors of the host. Do these small SNPs have anything to do with the lifestyle or the health of the host? How do we do that? For this study, we used a cohort of over 6,000 people who are generally healthy, and we have data of their health, of their lifestyle, different characterizations of them, and we also have their gut microbiome samples. The first step was to find these variable positions, the SNPs. So we compared the samples from different individuals. The first step is metagenomic sequencing, and then to compare different samples for the same genomic position for each bacteria, we aligned the samples, the short reads, to known bacterial genomes. We compared the reads that came from different samples to look for variable positions, which we called SNPs. The second step after finding the variable positions was determining finding the major allele, the cohort-wide major allele of each position, the most frequent allele for this genomic position of the bacteria. And then, this is here in the middle. And then after we have the major position, the major allele for each position, this is one bacterial species, for example, in each of its variable positions, we wanted to quantify the major allele fraction of each sample. So this is the genotype of each sample. The genotype of each bacterial position in each sample is a fraction that's between 0 and 1 that quantifies the fraction of the major allele out of all the reads that we found that aligns to these bacterial positions. So in human genetics, when we look at one SNP and one participant, this participant may have one copy of the major allele, two copies of the major allele, or no copy of the major allele. But of course, when we look at the microbiome, a participant may have 700 copies of the major allele and 400 copies of the minor allele, and any fraction can exist. So we quantified it with a continuous number between 0 and 1. So the question we asked before is now whether these vectors of the MAF, the major allele fraction of each position, and the vector of the phenotype of each participant correlate. To assess the correlation, we use the regression model to add also other covariates, because if, for example, we wanted to test the association between a SNP, a single SNP, and the age of the host, we wanted to make sure that we don't find SNPs that correlate with the BMI of the host, because BMI and age are correlated in the cohort. Okay, so what did we find? First of all, we found around 20 million variable positions in 578 bacterial species. So this is the number of hypotheses we have for each of these SNPs. Now we want to ask whether this variability in this one single position in the bacterial genome somehow associates with the health of the host. And in this proof of concept study, we studied the associations between SNP and 4 host phenotypes. And we asked whether SNPs associated with host age, whether they associate with host sex, BMI, body mass index, and percentage of glycated hemoglobin, which is a marker for diabetes. And what we found was that many bacterial SNPs associate with each of these phenotypes. For example, these are the results of the associations between bacterial species and host BMI. So each dot here is a SNP, a bacterial SNP, and they're sorted along the x-axis according to their taxonomy. And on the y-axis, you see the p-value of the association between this particular SNP and the BMI of the host. So the higher the point is, the more statistically significant was the association, and everything that's above the red line is more significant. It passed the Bonferroni correction for this number of hypotheses. And for some of these top SNPs, you also see the gene tag, if they fall within bacterial genomes, the gene tag of the bacterial gene. So you see that these associations come from the entire spectrum of taxonomic range within the microbiome samples. And so these SNPs come from different bacteria, and around 10,000 of them were significantly associated with host BMI. These are the associations between bacterial SNPs and host age. We found even more associations with age than we find with the other phenotypes. You can also see in this plot for each SNP whether it falls within a gene, outside the gene, whether it's predicted to be synonymous or non-synonymous, or whether it's a known protein coding gene. And most of the top hits here are within genes. These are the associations between bacterial SNPs and host sex. And then the associations with host hemoglobin A1c, and where we find the smallest number of associations, although this was a cohort of healthy participants without diabetes, and the range of hemoglobin A1c here wasn't very large to begin with. Okay, next we wanted to know whether the associations we found are random only because we tested for 20 million SNPs. Something must come up even randomly. So just to make sure that this wasn't random, we also conducted a permutation test. We assigned each participant a random microbiome samples from the pool of samples, and then repeated this same analysis, and we did not find any statistically significant association. So everything I showed you before is probably not just random results from the large number of hypotheses, the structure of the data, or something like that. Okay, what we see here is for one SNP. This is the SNP that had the smallest p-value in the association with age. The right box plot shows the age distribution of people that had the alternative allele in their microbiome for this position. The middle box shows the distribution of ages for people who had the major-allele. And you see that there's an average 10 years difference between people with both, with the different bacterial alleles. And then on the left, you see people, the distribution of ages in people that did not have this species in their microbiome, or at least it wasn't detectable in their microbiome. And what you also see here is that the ages of people without this bacterial species and the ages of people with this bacterial species, but with the major, most popular allele of this species, is about the same. And this happened a lot. For each of the SNP, for each association that we found, we wanted to know whether the bacterial species from which the SNP came was also associated with the phenotype. What do I mean? So at the beginning, I said that a very common approach to microbiome studies is asking whether the relative abundance of a certain bacterial species associate with host phenotype. So we wanted to know whether the associations we found are in species that are also associated by their abundance with the phenotypes. And in most cases, in many cases they were, but in most cases they weren't. So if you look at the relative abundance of these species and ask whether it's associated with age or other phenotypes that we tested, in many of the cases, you'll think that this bacterial species is not associated with the phenotype. But when you look at these fine differences at the single nucleotide resolution, you see that there is some association. So this bacterial species is somehow associated with the phenotype, just not by its entire genome abundance, but maybe at the smaller difference. Okay, what you see in this plot is along the x-axis, each dot here again is a SNP that was tested for its association with host PMI. And along the x-axis, you see the average difference between people who had the major allele in their gut bacteria and people who had mostly the minor alternative allele in their gut bacteria. And these differences go up to over 3 BMI points, which is it's over 8 kilograms for someone who is 1.7 meters tall, which is quite a big difference. Now, of course, for all of these associations that we found between bacterial species and host phenotypes, we don't know what's the causality, what's happening here, whether these associations are the result of the SNP effect on the host phenotype, whether they are the result of the host phenotype or host lifestyle affecting the bacteria, and this is why this association rose, or something else. But in cases in which maybe sometimes the different bacterial functionality may affect the host health, you see here that the differences may be very big, potentially, because they go over 3 BMI points. And of course, if we'll find such SNPs that have such a big effect on the host BMI, it may be very interested to study further and maybe think about how you can use these SNPs in future interventions, whether by modifying the microbiome or just by giving this specific enzyme variant that's associated with the better phenotype or the product of this enzyme. But as I said, this association may be also the result of the host lifestyle or health somehow affecting the bacteria. And this is also interesting. So what you see here is for each of the, for each SNP we found to be associated with a phenotype, we also asked what gene do this SNP come from. Then we asked whether the associations also repeat in orthologous genes of different species. For example, if we found one age-associated SNP in one bacteria in the gene that codes for RCSC, do we find SNP that's associated with age in other species in these genes too? So as you see here, some of these gene groups represent many repetitions of ages of phenotype-associated SNPs in orthologous genes of different bacterial species. The top one being hypothetical protein, which is the most common gene annotation in our set. The second one, sensory histidine kinase RCSC, is a component of a response system of bacterial to external stressors. It codes for different functions that are related to cell wall synthesis, flagellal synthesis, and in E. coli, it was shown that the deletion of this gene in E. coli that reside in mice, they showed that the deletion of this gene in the E. coli made it not be able to colonize the mice gut anymore. So this gene may be associated somehow with the interactions with the host and colonization of the host gut. So maybe looking at these 40 different SNPs we found in this gene in different species and asking what site do they fall in, what's the effect of this polymorphism on the function of the gene, we may understand better the interactions between the bacteria and the host, and whether the direction of the causality is one or the other. Okay, so to summarize this part, we created a framework to systematically find SNPs in gut microbiome bacteria and then associate them with different host phenotypes. We found many statistically significant associations between bacterial SNPs and host age, sex, BMI, and hemoglobin A1C. And since each of these associations can be mapped back to a specific gene or a genomic region, we can suggest now various mechanistic hypotheses based on these associations. This is the research group of Professor Liron Zahavi, and Lee Reicher and Amit Levon from our group also contributed a lot to this study. And since we are done early, I'll be really happy to answer questions if you have some.
There is one question in the chat. Do you see it, or should I read it?
Yeah, wait, I'll open. Yeah.
Mm-hmm.
Okay. So there was a question: how deeply should the metagenomic data be sequenced to do SNP analysis? Thanks for the question. I did not mention it. So What we have in this cohort is about, on average, I think 10 million short reads per sample. This is the average in this cohort. This is before filtering for human DNA. And since we are relying on large numbers, we have, what I mean is that we have many samples here. We didn't ask for very deep coverage of each position within each sample. So in a sample, to call the allele of a certain genomic position, we could rely on one read, and it was enough for us because we assume that when looking on thousands of people, on average, we'll have good representation of whatever we have. Of course, the deeper you can sequence a sample, you'll have more of the variation because, as I said before, there is also a variation within a sample in a specific gene, in a specific SNP. I hope that answers the question.
Yes, thank you. So is this methodology published as a method as well, or it's in progress?
So we're now submitting this paper soon, hopefully. So I hope it will be published, not in too long. Yeah, but currently it's not published yet.
And how do you build this reference database necessary for this analysis?
Right. So we used along the years, we tried to use different reference data sets of bacterial genomes. The current one that we used was just assembled by our group and was also submitted for publication in the past month. So I hope it will be published soon and then everyone can use it. It's based on both samples from Israel. So they represent we hope that represent the same bacterial diversity that we have in this cohort, and also people from the US and different countries. I think mostly, if I remember correctly, Israel and the US. And also a combination of previously published data sets, reference sets and data sets. Yeah, the previous one that we used before that, which may be also useful for your analysis if you're doing something like that, was published by Pasoli et al. From Nicolas Segata group. It's also a pretty big reference set of bacterial genomes.
Okay, thank you. Does anyone else has some questions?
Have you looked at SNP association with the taxonomic composition itself?
Thank you for this question. No, we didn't do that, but that's something that we're really curious about, and it's probably one of the things we're going to do next. See whether it's interesting because, first of all, maybe one hypothesis that you may have, maybe this is why you asked it, is that the association between a SNP and the human phenotype is mediated by the SNP helping the bacteria to be more abundant